Hello everyone and welcome back to the Mobiochem Research Group. I'm Vito and today's lecture will be about molecular dynamic simulation of photoswitches inside of an ion channel. In particular, I will be talking about azobenzene and p-Diaminoazobenzene in the human's voltage-gated sodium channel. I will introduce you to this interesting field of study and talk about the enhanced sampling method and the free energy calculation performed during the experiment. Before we start, here are some references. The first one is our newly published paper, followed by a really important review on photoswitches in biological systems, and two papers which I used to learn about Gaussian-Accelerated Molecular Dynamics and end-state free energy calculation. The use of pharmacotherapies to treat both acute and chronic medical disorders has contributed to improve the quality of life in our society. However, many drug-related issues are still present in many therapies. Poor drug selectivity is one issue, which can lead to the evolution of pathogens due to the presence of the active drug throughout the full metabolic pathway. Light shows a large degree of orthogonality towards most biological systems, since photons with a wavelength within the therapeutic window do not contaminate the system and have low toxicity. This is why photopharmacology has been developed to design compounds called photoswitches, which undergo reversible changes in their structure and function upon light absorption, and control the function of biological systems with high precision. So, what are the criteria for a good photoswitch? First of all, it must be non-toxic before and after irradiation. Then it must absorb light above a certain wavelength in order to avoid tissue damage. Also, it must have a high efficiency of photoisomerization and, upon absorption, have a large structural change and be stable in biological conditions. But we will be talking about the photophysics of azobenzene in more details in the next video. In the last years, several photoswitches have been designed to modulate the activity of voltage-gated ion channels. Those transmembrane proteins play a crucial role for neural transmission, muscle contraction, and many more functions. Since action potentials are generated by the movement of ions across the membrane through the ion channels, the regulation of ion permeation by the combined action of light and a photoswitch is a potential tool to manipulate the function of voltage-gated ion channels with high spatiotemporal precision. What we see here is a simplified graphical representation of an ion channel inserted in a lipid bilayer. We see the selectivity filter, which is the narrowest part of the pore, the cavity, and the inner gate, and represented in blue the position of the photoswitches which will be inserted, azobenzene and p-Diaminoazobenzene. What we see here is a graph representing the activity of a drug throughout its pathway. A conventional drug remains active in its full evolution in the target, body, and environment. A prodrug, which is activated at the target, remains in its active state later on in the body and in the environment, while a photocontrol drug can be activated and also deactivated at the target. This effect can lower the toxicity and, as I said before, reduces the evolution of pathogens in the body and in the environment. Now let's get to the structure of the experiment. First, I will be talking about the preparation of the initial structure of the protein and the photoswitches, then introduce the production run performed with conventional MD to equilibrate the system, and Gaussian-Accelerated MD for the binding pocket search. Finally, I will talk about the evaluation of the binding free energy of each binding pocket with mmGBSA. So, let's start from the protein. The first thing was to obtain the structure of the human's brain sodium channel from the protein database with the following ID. The structure was resolved with cryo-electron microscopy in 2018. Then, with the structure obtained, the transmembrane proteins was amidated, acetylated, and placed in a POPC lipid bilayer with Charm-Gui bilayer builder. Then, finally, regarding the force field, the potential parameters of the lipids and the proteins were taken from the Charm-36M, then TP3P was used for water, and the Lennard-Jones potential parameters for the ions were adopted from these two authors. The two photoswitches, azobenzene and p-Diaminoazobenzene, were first built with Charm-Gui ligand reader and modeler. Then the geometry was optimized at the MP2-631G star level of theory, And then, in order to obtain good atomic charges, the restrained electrostatic potential charges were obtained at the HF631G star level of theory. And now let's go to the simulation details. In the first stage, we run 150 nanoseconds of classical MD without the ligands in order to just equilibrate the protein. Then 200 nanoseconds of classical MD were run but with each ligand inserted to equilibrate the full system. At this point, 4 replicas of 1000 nanoseconds of Gaussian-Accelerated MD were run with each ligand in order to find the binding pockets. And then, going back to classical MD, we run 100 nanoseconds for each binding pocket found in order to confirm the stability of the ligand. A great challenge in molecular dynamics comes when regions of high probability in the phase space are separated by large energy barriers, where it is very unlikely that all important configurations are sampled unless infinite simulation time is used. This is known as the problem of quasi-non-ergodicity. Now, in the lack of predefined reaction coordinates and when all important regions of the phase space are not known, a good method which can be used is Gaussian-Accelerated Molecular Dynamics. The method adds an harmonic boost potential to the full potential energy surface, below a certain threshold energy. And this is done in order to accelerate the transitions between low energy states and becomes beneficial in our case for free drug binding processes, reducing the computational cost of the simulation. Two reaction coordinates were chosen to calculate a free energy surface which can give an indication of the regions of highest probability of the ligand inside of the channel. To do so, the first stage was to calculate the center of mass of the alpha carbons of the 4 residues composing the selectivity filter, and the 4 residues composing the gate, as well as the center of mass of the ligand throughout the full 4 microseconds of simulation per each ligand. Then, the two reaction coordinates were chosen to be the distance between the gate and the ligand, and the dot product between the two vectors going from the selectivity filter to the gate and from the ligand to the gate. Then, finally, the bias free energy surface was reweighted along these two coordinates, with pi reweighting toolkit with a 10th order Maclaurin series. The free energy of binding was evaluated with MMGBSA, Molecular Mechanics Generalized Bourne Surface Area. This is an end-state method which could be placed in the center of a spectrum of precision and computational cost, where in one end we see the scoring function of molecular docking, and on the other side we see the alchemical perturbation methods. This is because the method is only based on the solvated complex trajectory, also called the end state. The method allows to find the difference in binding free energy computed as the energy of the solvated complex minus the energy of the solvated receptor minus the energy of the solvated ligand. Here on the left we see that we start with the complex, we run an MD simulation with the explicit solvent and we obtain the trajectory for the complex. Then the script removes the solvent molecules and inserts an implicit solvation based on the generalized Born equations, and then computes the terms in such a way that as we see here, the angle brackets are based on the ensemble of the complex trajectory. The binding free energy is obtained from an enthalpic and an entropic term. The enthalpic term is approximated into two terms, the first one being the interaction between the ligand and the receptor, and the second one being the interaction of the ligand and the receptor with the implicit solvent. So the first one is obtained from the force field describing the bonding energy, so the distance, angle, and dihedrals, the electrostatic interaction, and the van der Waals interactions between the ligand and the receptor, while the solvation term is divided into a polar part, which is described by the generalized Born model, and a non-polar part, which is obtained from a linear relation to the solvent accessible surface area. In the simulation, it is assumed that the change in entropy of the different ligand poses inside of the binding pockets is equal to 0. Therefore, the entropic term can be discarded. Also, in the 1 average MMGBSA, the bonding interaction are equal to 0, because we're only considering the complex trajectory. With this being said, equation 7 now reduces to the following equation 9, including only 4 terms. Therefore, the total binding free energy can be rewritten as in equation 10. Also, another important part of MMGBSA is that each term can be pairwise decomposed, which means that we can actually find each of those terms, being the electrostatic and the van der Waals interaction and the polar and nonpolar solvation energies, between each residue and the ligand. Therefore, we can understand which are the most contributors to the binding of the ligand in that binding pocket. Now let's get to the results. I will first introduce the equilibration stage, showing the RMSD of the protein and the ligands. Then the binding pocket search with the free energy surface of the two reaction coordinates chosen, and finally the MMGBSA results with the total binding free energy and the pairwise decomposition analysis. This plot represents the root mean square deviation of the ion channel with respect to its first aligned frame, removing the translational and the rotational motion. We see in black the first 150 nanoseconds of simulations without the ligand inserted, and we see that approximately after 50 nanoseconds the structure is equilibrated, while in blue and green we see the following 200 nanoseconds of simulation with the ligands inserted. Also here we see that after 100 nanoseconds the structure of the protein is equilibrated, and therefore it's possible to run Gaussian-Accelerated MD starting from the last snapshot of the trajectory. To refresh the memory, as I said before, two reaction coordinates were used, the first one being the distance between the gate and the ligand, and the second one being the angle between the two vectors, going from the selectivity filter to the gate and from the ligand to the gate. The two reaction coordinates were then used to reweight the bias potential and obtain those two energy maps. Those energy maps represent the position of azobenzene and p-Diaminoazobenzene inside of the channel. It is important to consider that this is only a two-dimensional cut of the multidimensional potential energy surface, and that the y-axis, representing the angle, could have the same value for the ligand being in the left or in the right side of the cavity. Therefore, it is also important to compensate with visualization before drawing any conclusion. For azobenzene, we see four distinct pockets, with the fourth one being the least visited, while for p-Diaminoazobenzene, we see five different pockets, with P1 being the one with the highest probability distribution. This first image shows one representative snapshot for each of the binding pockets for azobenzene, showing with different colors the poses of the ligand and the four domains of the channel. The P2 pocket is the closest to the selectivity filter in the central cavity of the channel. The same image is now shown for the diamino derivative of azobenzene, and in this case the binding pocket closest to the selectivity filter is pocket 1, P1, which is also the most visited point for the ligand throughout the 4 microseconds. The two pockets I've shown here and in the previous slide are the ones with the highest binding free energy, as we will see in the following slides. With the free energy surfaces, the visualization and the root mean square deviation of the ligand with respect to the line proteins, it is now possible to identify the different pockets. An intuitive relation between the energy map and the root mean square deviation can be observed in replica number 4 in the bottom right. We see that the ligand is being stable in the pocket 1, P1, throughout the full microsecond, and this can also be observed in the energy map, where P1 is shown, where it has the highest probability distribution. Now let's go to the results of the binding free energy. First of all, 100 nanoseconds of conversion MD were run for each pocket. This was performed to confirm the stability of the ligand and to obtain unbiased energy terms. Then, before running the MMGBSA and the pairwise decomposition analysis, the total binding free energy of p-Diaminoazobenzene was computed with a different amount of frames and a different time length. From the graph, we can observe that the difference in energy between 100 and 1000 frames is only 0.2 kcal per mole, and also that the time length of the simulation does not affect the weight of the computation in MMGBSA. Therefore, it was chosen to take 100 equidistant frames into the 100 nanoseconds in order to capture the full dynamics of the simulation. First of all, 3 of the 100 nanosecond simulations were discarded, since the ligand escaped from the binding pocket. Then, now, from the table, we can observe that the highest binding free energy for azobenzene is in pocket 2, while in the case of p-diaminoazobenzene is in pocket 1. Both of those two pockets are close to the selectivity filter, which is in agreement with previous docking simulation and X-ray structural data for quaternary ammonium ligand interacting with the central cavity of the potassium channel. This type of binding, close to the selectivity filter, leads to the direct inhibition of the channel by blocking the pore. Also, the binding of p-diaminoazobenzene to the protein is stronger than the binding of azobenzene, not only because of the size, which leads to a larger number of interatomic interactions, but because these interactions are stronger. If we normalize the binding energy per atom, p-diaminoazobenzene has a larger amount of kcal per mole. Also, the table shows that the most important contributions are the van der Waals interactions between the ligand and the protein. Also, the unfavorable polar solvation energy means that the electrostatic interactions between the complex and the solvent are less attractive than the interactions of the individuals with the solvent, while the opposite happens for the non-polar solvation energy. Finally, the electrostatic interactions between the ligand and the protein are more important for p-diaminoazobenzene than azobenzene, which we will see later are responsible for the larger total binding free energy. To gain more insight into the nature of the ligand-protein interactions, the binding free energy for the 3 more stable pockets of azobenzene and p-diaminoazobenzene were decomposed into pairwise ligand residue energies. The photoswitch, as shown here in the figure, undergoes the strongest interaction with non-polar residues. For example, the strongest contribution to the total binding free energy comes from phenylalanine 1243, which interacts with azobenzene by means of stacking interactions. In the case of p-diaminoazobenzene, the ligand-protein interaction is also stabilized by non-polar amino acids. The importance of non-polar proteins has also been found for the tetrabutylammonium ligand bind to the KCSA potassium channel, for which the ligand was stabilized by interaction with the side chains of phenylalanine. Now, despite the importance of hydrophobic interactions, the photoswitch undergoes intermolecular interaction also with polar residues, and here in the image we see serine 1585. The visualization of the dynamics also reveals that part of the electrostatic energy is caused by H-bonding, where one of the amino groups of p-diaminoazobenzene acts as a hydrogen donor, and the AOH group of serine 1585 acts as hydrogen acceptor. For the H-bond analysis, it was assumed that the H-bond is formed when the distance between the hydrogen donor and the hydrogen acceptor is shorter than 3 Armstrongs, and the angle between the donor, the hydrogen, and the acceptor is larger than 135 degrees. The hydrogen bond between p-diaminoazobenzene and serine 1585 for P1 shown here is present along 42% of the simulation time. Now to conclude, molecular photoswitches have been developed over the last decades in the field of photopharmacology to enhance the selectivity of medical treatments with the help of this high spatiotemporal resolution that can be achieved with light. In this work, the binding of azobenzene and p-diaminoazobenzene was simulated with an enhanced sampling method called Gaussian-Accelerated Molecular Dynamics, and the binding free energy was computed with 1A mmGBSA. First, 3 pockets were found for azobenzene, 2 of those close to the selectivity filter, and the binding of azobenzene, as we saw before, was largely controlled by hydrophobic interaction. 3 stable pockets were identified for p-diaminoazobenzene, where the most stable was also found inside of the central cavity of the channel, close to the selectivity filter. Also, in this case, the binding is controlled by van der Waals interaction, but is also stabilized by polar interaction with polar residues. In the case of the most stable pocket, a large fraction of this electrostatic energy is caused by hydrogen bonding. between the ligand and serine 1585. So overall, p-Diaminoazobenzene is able to bind to the channel stronger than azobenzene. The presence of different binding pockets does not imply that the ligand will block the transport of ions through the protein, but it is promising that the binding occurs close to the selectivity filter. Therefore, we will need to continue research to elucidate the mechanism. Also, the next step will be to investigate the effect of the protein environment on the electronic structure and the photophysics of those photoswitches.
So this is the end of the video. Thank you very much for your attention, and I really hope you enjoyed it.